students in the audience, in addition to being inspired by the phenomenal lecturing abilities of the speakers and the exciting research that they're doing, are also paying attention to the diversity of career paths that our lecturers have taken, from starting from card-carrying physicists to work on a great diversity of problems that are of importance in science. And our next speaker is no exception to that. Dave Weitz from Harvard is the Mallinckrodt Professor of Physics and Applied Physics and Professor of Systems Biology at Harvard. He's the co-director of the BASF Advanced Research Initiative at Harvard and the co-director of the Harvard Kavli Institute for Bionanoscience and Technology. He's director of the Harvard MRSEC. He's best known for his work in areas of diffusing wave spectroscopy, microrheology, microfluidics, rheology, fluid mechanics, interface and colloid science, colloid chemistry, biophysics, complex fluids, soft condensed matter physics, phase transitions, the study of gases and amorphous solids, liquid crystal self-assembly, and surface-enhanced light scattering, and diffusion-limited aggregation. That was all from his wiki page. Dave began his career at Exxon Research, and he worked there for nearly 18 years. Then he became a professor of physics at Penn and then moved to Harvard in 1999. Dave's a member of the National Academy of Sciences, the National Academy of Engineering, the American Academy of Arts and Sciences, and he's a fellow of the American Physical Society. I hope that some of you came to his outreach session on Monday, where he teamed up with Chef Spike from Woodbury Kitchen in Baltimore to teach us about the soft matter physics of cooking. And now, Dave, the stage is yours.
As usual, Sharon knows more about me than I do. Thank you, Sharon. Also, thank you for the invitation. It's really a pleasure to be here. I think I'm going to change the theme a little bit from what we've heard, just spectacular talks. I'm going to talk about something that I hope has really, well, for me, has really interesting physics, but also has a lot of practical applications. And I want to share both of them with you. So I'm going to talk about new ways of making materials. It's a way that when I first saw it, I didn't believe you could possibly do it. But over the years, we've learned how to do these things, and we've been able to make really remarkable, or at least to me, remarkable materials. And they have a lot of interesting physics. Understanding the physics of how they work, at least for me, has been a lot of fun. But they also have a lot of practical uses, and I'd like to try and share both of them with you. So let me say at the beginning that all of the work is that of a very talented group of postdocs, graduate students, visitors who have worked with us, as well as some wonderful collaborations with several of my colleagues at Harvard. So the way we do everything, and what I'm going to try to describe to you, is a very simple way of making particles, but taking advantage of making a single drop of one fluid in a second fluid. The beauty of that is that the surface tension keeps the drop really well defined. It keeps it spherical. And you can use this not only as something to contain things, but something to build things around it. So you can use the droplets to make larger, more complex, and more intricate structures. You can make them very, very uniform, very absolutely the same size. You can use each drop. You can fabricate and synthesize new kinds of materials, but you're doing everything one by one. That has the tremendous advantage that everything's identical. Everything's exactly the same. Of course, it has a small disadvantage that can you really make sufficient quantity to make something useful? Well, we do it, to do this, I'm going to try and answer all these questions to show that you can do all of these things, including making things useful. We do it by taking advantage of microfluidic techniques. And to me, microfluidics are nothing more than making structures, making three-dimensional structures using whatever technology you have available that have the size of the fluids that you want and allow you to mix the fluids in exactly the precise way that you want. So how do you make drops? Well, drops are an emulsion. They're two immiscible fluids. So here's one of my fabulous students, Anderson Shum, who's now a professor in Hong Kong. He drew a little picture. How do you mix oil and water, oil and vinegar if you're like a vinaigrette for your salad? How do you do it? you shake, right? There you go. That's shaking for you, Sharon. You get something like this. You get drops, but they're not very monodisperse. What we want to do is make absolutely precise drops, absolutely, completely uniform drops. And to do this, we developed over the years a new kind of microfluidics, or a separate kind of microfluidics, very, very simple microfluidics, just takes advantage of capillary tubes. Capillary tubes are used all the time by biologists for patch clamping, for all sorts of things. And the nice thing about a capillary tube is you can start with a capillary tube that has a diameter of about a millimeter, and just by heating it and pulling it, you can sharpen it down till it has an orifice, a small opening that can be as small as a micron. It's a very easy and very reproducible thing that you can do. Now, to actually design and organize these capillary tubes, we used, again, something very, very simple. We took advantage of the fact that the outer diameter of the capillaries are also absolutely well-defined, and they're cylindrical. So if you take a square capillary, which you can get, and you thread the cylinder through this, and of course you adjust the dimensions here so that they're the same, then you can thread it and align it in a very, very simple fashion. And so you can build something quite simply, and there are 4 little interstices here where outer fluid can flow, and since it's neck down, it is a small orifice, the flow rates are roughly comparable. And so you can have flow in the same direction in a very simple fashion. Here are the square capillary, the cylindrical capillary. They're put together like this, and they're all held together with our favorite ingredient, just 5-minute epoxy. You can build a device like this really very, very quickly. Anderson could build a device like this easily, even a much more complicated one, in half an hour. And they work really well. Here we're injecting water into oil or oil into water, it doesn't matter which, and you can see that you get perfectly uniform drops. Oops, sorry. You get perfectly uniform drops. They're all exactly the same. And this should remind you of something. It should remind you of something going drip, 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 drip. But if you increase the flow rate of the drops, you still get drops, but now you get a jet that's formed. And if I try to put this laser pointer where the drops form, you can see sometimes they form right where the laser pointer is, but they wander a bit. So when you have this jetting phenomena, the drops are not quite the same, quite as uniform in size. Actually, we can understand this. Oh, did I tell you that you have a homework problem? You actually have to go home and do an experiment. You can do this experiment really easily. Go home and turn on the faucet in your kitchen, and turn it on really, really slowly, and you'll see it go drip, 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 always drips. What's happening? It's a Rayleigh plateau instability that's causing the drops that form. But if you turn the water on just a little bit harder, you start forming a jet. But if you look very carefully and you adjust it precisely, you can see just before it hits the sink on the bottom, it breaks into drops. And if you can't do it, if you can't adjust your faucet, go and take a water hose, something that sprays a huge amount of water, but spray it as far as you can, and you'll see it always breaks into drops. It breaks into drops because the surface tension always wants to make the fluid into a cylinder of fluid into a drop. We can understand the dripping very easily. You can do this experiment yourself. If you turn the water on very, very slowly, then the force of surface tension holds the drop in place. And as it grows, its weight gets larger. It gets larger and larger until finally the weight of the drop exactly balances the force of surface tension, and then it breaks. But since it's just a balance of forces, if you have a clean faucet, it's always exactly the same size. In the case of the jetting, it's just a Rayleigh plateau instability. It's just saying, well, anytime you have a small indentation, a small fluctuation, the Laplace pressure, which is inversely dependent on the radius, the Laplace pressure gets larger and it drives the fluid on either side. In fact, we can calculate that. We can calculate the time that it takes to move in this direction just by saying, well, look, it's being driven by the Laplace pressure, so it's being driven by surface tension, it's being resisted by viscosity, so the velocity of motion in this direction is just this ratio. And then you can calculate the snap-off time just from the ratio and knowing the radius of the cylinder. So now we can actually understand jetting in a very simple way. Now what happens is if you have a fluctuation and it's moving downstream at the same time, we know the snap-off time, we know the velocity, and so we can calculate the length of the jet. And then if we want to ask where does the transition between jetting and dripping, we just have to say, well, I have to fill the drop to its size, and so I make the length of this jet exactly equal to the drop size. And that turns out to be at a capillary number of 1. A capillary number is just this dimensionless number, depends on viscosity and surface tension. There is actually another kind of instability. This is what happens if you flow the fluid much more rapidly. Here there's some particles. You can see they're moving very rapidly through the into the drop to show that the fluid is flowing rapidly. This depends on the inertial effects and therefore on a Weber number. But you can put everything together into one phase diagram that just depends on the sum of these two dimensionless numbers, and you can determine where you get dripping and where you get jetting. So you can understand the behavior fairly well. But we can do a lot more because we can organize these capillaries in a very simple way. So this is the first thing. In fact, one of my students, Andy Utada, came, and he was a great student. He made lots of mistakes. So the first thing he did was he hooked everything up wrong, and everything was backwards. So instead of having the two fluids flowing in the same direction, he had one flowing the other direction. Actually, this works pretty well. You just get hydrodynamic flow focusing of one fluid into a second. But what's really cool here is you have this extra room. You can add a second capillary. It's very easy to align. You can add the second capillary. And now you have flow focusing of a co-flow in this side and being flow focused. And so here's an example, and you can see these drops, you have drops inside of drops. Actually, maybe it's better to look at this example where by changing the flow rates here, by changing the flow rates, you can see you get always, in all cases, you have a multiple emulsion, a drop inside of another drop, but just by changing the relative flow rates, you can get small drops and large drops, or you can make the shells of different thicknesses. Already, this becomes very interesting. You can make interesting materials with this. Let me give you a few examples. Let me imagine that I have a material where I make drops. I make this double emulsion where the outer fluid and the innermost fluid are the same. Let's say they're water. And I use as the shell, I use some solvent. And I can put some surfactant, some phospholipids, some diblock copolymers, something that's amphiphilic, something that will go to the surface. Those molecules will coat this surface and this surface, both of them. But if the solvent then can be evaporated, if it's volatile, I can evaporate it. What am I left with? I'm left with a bilayer structure. But what's interesting about that bilayer structure is that the inside fluid and the outside fluid are completely different. So if I want to encapsulate something, such as in a liposome or in a vesicle or something like that, I can do it in a very simple fashion. Actually, it's interesting to watch the evaporation. Here's a movie of watching the evaporation of the drops. You see what happens. This is actually the drop. And what you notice is that there's actually a contact angle. There's a surface tension, a contact line, a wetting. So you can only have this contact angle if there's some adhesion between the two shells, the two layers of the shell. So they're actually slightly adhesive. And in fact, you can do some experiments, and this is something that Anderson did, you can do some experiments to vary the contact angle, and you find that when the contact angle is 90 degrees, which is the maximum it can be, you get the most stable drops. Now, just to show that these are really flexible or hollow shells, I'd like to show that you can collapse them. Well, how do I collapse them? I push on them. How do I push on them? Well, I can't really put hydrostatic pressure on. That won't do anything. But I can put an osmotic pressure on. So I put something that will draw the water out. And here's some images of what happens. And you can see that, in fact, what you see is that they buckle. They can only buckle if they're an elastic shell. And in fact, Sujit Datta worked with my colleague David Nelson. And by measuring the pressure, you're just applying a pressure. By measuring the pressure it takes to buckle these things, you can actually measure the elastic constant of the shell. Actually, if you press on them, you can squeeze them. And look what happens as you squeeze them. You can squeeze them so they get smaller and smaller and smaller until finally you've squeezed everything out. Well, if there's something inside, I've just showed you now how to get rid of it, how to make it escape. So if I've used it to encapsulate something, I've showed you how to release it. You can do another thing. You can put it into osmotic shock. That is, you can blow it up. Basically, there's some osmotic pressure on the inside. If you put it into pure water, there's no osmotic pressure on the outside, and it blows up. Here's a case where we have some dye molecules inside. We put it into osmotic shock. I would show it to you later with the dye molecules, but they're none there. They've all diffused away. Actually, it's interesting to watch. Watch this particular drop. As we put it under shock, you see that first it grows. Water is flowing in, and then a little hole breaks, and it starts to get smaller. Same thing here. In this case, it completely disappears. You can do a lot of other things. So one thing you can do is make shells of liquid crystal. You make them isotropic to make the emulsion, and then using chloroform, you evaporate the chloroform and make it into a liquid crystal. So Al Fernandez came along, and he was challenged by my colleague David Nelson, who really wanted to see a thin shell of a liquid crystal because he said there should be 4 defects. And sure enough, Al found 4 defects in it. But then he did a few more experiments, and he found that sometimes you saw 2 and sometimes you saw 3. And so this led to a lot of really interesting work that he could do to try to understand the defect structure of thin shells of liquid crystal. Liangyan Chu came and visited us from Sichuan University, and he's working with Andy, and he designed a slightly different way of making these double emulsions. In this case, he used 2 co-flow devices, one, and then that leads into another. And if you watch here you have 1, 2, 3 drops, and it snaps off. The nice thing about this is you have perfect control of all the flow. And so now you can make what I call designer emulsions. Now if you say you want 5 drops, I say, well, that's not enough. Tell me what size you want. Do you want small, large, or medium? You can change both the size and the number in a very easy fashion. However, what's really interesting is, look, here's 1, here's 2. What about this? Why don't we just add a 3rd? And so now you can make real Russian dolls of drops, drops inside of drops inside of drops in a very, very simple fashion. Of course, you look at that and you say, well, that's great. That's a wonderful academic exercise. Could it possibly be of any use? And actually it is. Here's an example. This is a case of an emulsion. It's a water and oil emulsion, and it's surrounded by a hydrogel. And it's all in an oil base. This is actually a very good way of encapsulating something. For example, if you want to encapsulate a very delicate peptide in the presence of a lot of surfactant, you can do this. And the hydrogel is perfect encapsulant. This hydrogel, this particular hydrogel, is thermosensitive. So as you heat it up, it starts to shrink. And as it shrinks, oops, didn't want to play. Let's see if it does. As it shrinks, it drives out the water. It can't compress the inside, and eventually it tears itself apart and releases what's inside. So this is actually a very good way to build a structure that's really good for encapsulation. It's very robust encapsulation, but you also can control the release. And that's actually very difficult to do with many structures. You can do a lot of other things. Shin-Yun Kim came to the lab, and he found a way of coating the surfaces of the glass capillaries to put very, very thin layers of fluids, and you can make a very thin shell. Laura Arriaga used this to make real vesicles. And vesicles are actually very difficult to make with a thicker layer, but with the thin layers, they're very easy to make. Here's some large-scale vesicles. You can see the thermal fluctuations. But with Tina Huang, one of our graduate students, she's doing something a little bit different. She's adding not just a double emulsion, but a triple emulsion. But now there are 2 layers of oil, and each type of oil has a different phospholipid molecule in it. And then most of the phospholipids will go to the closest interface, and when it's evaporated, you actually get an asymmetric vesicle. That is a vesicle that has one outer leaflet with one type of phospholipid, an inner leaflet with a second type of phospholipid. This actually is very important for mimicking the behavior of cells, but is very difficult to make under normal circumstances. In this case, you get something like 75% asymmetry, which is consistent with just the diffusion of the molecules through the different thicknesses of oils. And so I think we could improve that by varying the thickness of the oil. So there are other ways of making the devices. We don't have to make them using glass. In fact, if you're at Harvard, what you do is you go across the street, you go to your colleague George Weitz, says, George, please show us how to use soft lithography. And he does, and we find out simple ways of making other types of devices. There's a lot of advantages to these kind of devices. Here's a double emulsion generator that Adam Abate made, and he shows you can easily make double emulsions. Just as easily you can make triple emulsions, you just stack them together. There's actually very, very interesting physics that everything is perfectly triggered, so you only get one drop at a time, and that's a very robust phenomenon. And so you can see there's triple emulsions. But Adam said, well, look, Wang Yin had already made triple emulsions. He's got to do something much better, so he made a quintuple emulsion. A drop inside of a drop, inside of a drop, inside of a drop, inside of a drop. You just stack them up, right? It's really easy. Esther Amstead came and she said, well, look, everybody's making drops in a fluid. What about making drops where one of the fluids is air? So she tried to basically make a spray dryer. She did this, so this is an emulsion generator, and this is now not a second fluid, but air. And she tried to do this, and she saw that she got a jet forming. She got another jet. She added more outlets or more inlets for air, and you always got jetting. And she said, well, look, we know that we can go between jetting and dripping. Can I make some drops in air? And to do that, she went back to what we calculated about this. And in order to do that, if you want to make a drop, you have to have a stationary instability. You have to have the velocity of the fluid in this direction equal to 0. But how can you do that when you're flowing air? Because air is flowing so rapidly. Well, she came up with a really trivial and simple and very elegant way. And she realized that only the velocity in this direction has to be 0. So she put the air in the opposite direction. It has to at some point turn around. So at some point there has to be a stagnation point. At some point it has to go to 0. And then she did the same thing, and she added more and more channels until finally you can see that you really can make drops. Now this is a dripping phenomena in air. But what's cool about this, you can measure the velocities of the air, you measure the velocities of the particles. The particles are about the same size of the channels. And what you realize is the air is moving much, much, much faster than the drop. Moreover, there's about a gap. There's a gap of about 1 micron. So the air is blasting by this drop through a 1 micron gap. And if you calculate the velocity of the air that it must be going, it's something like 60 kilometers per second. It's moving at about Mach 180. Moving out. Can I use normal fluid mechanics for that? I don't know. But let me just try and be very naive and do that and say, well, I have this viscous force through the flow of the air across the drop, and I have a surface tension that's holding it together, but the viscous force is so large that it's going to start breaking the drop into small drops. And I just take the balance. I say, well, this should actually break this large drop, 1 micron drop, into tiny drops, about 300 nanometers. And in fact, what you can do is you can put some material inside at a known concentration, and you can make a spray dryer. If there's 300 nanometer drops with the concentration of material we had, you should get about 30 nanometer particles, and that's exactly what you find. So this is a really wonderful way of making nanoparticles. And you can change the size of the particle just by changing the velocity, changing the pressure, you change the velocity in a completely predictable way. And you can make nanoparticles of almost any material. There's only 1 or 2 materials, in fact, that we've tried that you can't make nanoparticles. It's a very, very simple way of making nanoparticles. But what's also really cool about these things is that you can look at the structure. Most of the materials that I showed here all are crystalline. And if you make larger particles, you indeed see crystalline lattices. You see either single or multiple crystals. But if you make the particles small enough, you completely lose any evidence of crystallinity. The particles are completely amorphous. And you can do this is done with TEM, you can do it with X-ray scattering, you can do it with DSC. Here's some DSC curves. You completely lose all evidence of crystallinity. You make amorphous structures. Now, the reason for this we understand. It's a little bit complicated. I won't go through the details, but things really want to crystallize pretty rapidly. But what happens even before crystallization in this case is you get spinodal decomposition. And so you get a region with high concentration solute region with low concentration solute. The high concentration solute is so high in concentration that the diffusion of the molecules is slowed down. It becomes a glass and it essentially freezes in place. The low concentration area, the crystallization takes too long, so there's no chance for a crystal to form. So you're getting a frozen, spinodally decomposed structure. It's very similar to what you see actually with colloidal gelation. You see exactly the same kind of behavior. And we can predict this and measure this. Everything is consistent with that picture. What's also interesting is Esther measured some X-ray scattering. She looked at after some period of time. She made the particles. She came back and looked at 7 months later, and she found that they remained amorphous. So these things, even though they're amorphous and they want to be crystalline, they remain amorphous. And she did even more experiments, heated them up, and showed that they're very, very stable. The reason they're so stable, of course, is the fact that they're nanoparticles. If I wait until one of them crystallizes, they will crystallize eventually. Only that particle crystallizes. I have to wait till crystal forms in every particle. And that's kinetically stabilizing them. That will take a very, very long time. So these are very stable particles. That, of course, is really important for bioavailability for drugs. You want to have small amorphous nanoparticles. The solubility of something in the amorphous state can be a factor of 100 times larger than the solubility of something in the crystalline state. So Sharon said we often do things with food. What's the most common material that you have on your table every day? Is table salt. It's absolutely crystalline. You've never seen table salt that's not crystalline. But if you spray dry table salt, if you spray dry sodium chloride and make particles less than 5 nanometers, they're amorphous. So you can make amorphous table salt. You can make almost anything amorphous this way. In fact, just to prove that it's amorphous, we took the electron beam and we heated up the particle and showed that it crystallized back into salt. Well, you know, I told you that we're making particles one by one. You can see we have this elegant way to make very, very uniform particles. But the question is, can we make something useful? So when I first started doing this, my former colleague, Howard Stone, was also working on this, and he was supported by a large home and personal care company interested in seeing whether they could actually make things in a practical way. And after a few years of very, very good work, they decided, no, you can't. And when I heard that, I said, well, that's a perfect problem for Harvard students. Can you make something practical with this? Well, if you want to do this and you want to make large numbers, you can't just make a single device. You have to make many devices. So you have to scale it up. You have to parallelize. So you can go into 2 dimensions. You really want to go into 3 dimensions, because if I want to ask how much material I can make, I want to have a big volume of things I want to make material. So Mark Romanowski came and joined the group. He came to me, he said, you know, I've worked for 4 years, I have no results. I said, well, what did you do? He told me what I did. I said, well, why don't you, here's a challenge for you. I'll give you a liter that's 10 by 10 by 10 centimeters. You can fill it with devices. I'll give you 3 pumps, and I want you to make a device that will produce a liter of internal phase an hour. If I can make a liter an hour in this size, I can stack the room with these things. I can make reasonable quantity of materials. I said, if you can do this, you'll publish a good paper. He did publish a paper. It was the most cited paper of the month that he published it. I said, you also can graduate. He graduated. He's working, he graduated several years ago. He did this very nicely. This is a view of the device. You can see it's very small. This particular device has 25 emulsion generators. You can see that they're collecting here. Esther came along and she said, well, let's really go into much higher level of production. And she made what she called a millipede device. It's the millipede device because it's got 1,000 emulsion generators here and 1,000 emulsion generators here. And what's quite interesting about this device is that it's a completely different hydrodynamic instability. We had to go to our colleague Michael Brenner, who calculated the hydrodynamic instability. The beauty of this hydrodynamic instability is it's completely device dependent. It doesn't depend on flow rates. And so this is just an example. I don't have the time to tell you about the hydrodynamic instability, but it's completely determined by the shape of the device. As a result, it's very robust. You can vary the flow rate by enormous amounts, and the size of the drops remains exactly the same. If one of the channels clogs, you don't care. Everything remains exactly the same. So now this really starts to make large quantities of material. This is one device just looking down. You can see you're making quite a large amount of material. You can make different sizes. You change the device to make different sizes. But let's look how much material we're actually making. With this particular device, this is actually, the slide is a little old. Esther's done better than this since. She was getting 25 milliliters per hour of internal phase with one device. This is the size of the device. So all I'm going to do is just pack these without doing anything, without doing any kind of scaling. I just pack these to my canonical liter, 10 by 10 centimeters. That's 400 devices. So, whoops, this device produces 10 liters per 10 by 10 centimeters per hour. So now I can really produce a reasonable quantity of material, but I have to prove it to you. So with a couple of friends, we started a small company. It was called Capillary. We had to find a way of producing something. Drugs would be ideal, but it takes a long time to prove that you can commercialize it with drugs. So we had to find the next highest value-added material. Anybody know what that is? Somebody pay a lot of money for something. Cosmetics, right? So, okay, so Capsa made cosmetics. It was on the cover of CNE News. This is one of the emulsion generators, a more advanced one. This is one of the cosmetics. I've learned a lot about cosmetics. I guess half of you know something about it. I didn't when I started. This is made by La Prairie, but this is actually made with microfluidic techniques. I would actually show you one of these, but let me show you why it says value added. Because if you look there and ask what the price is, it's $500 for 50 milliliters. Actually, oops, let me show you this. Actually, you can make a lot of really interesting materials. Because you have the drops completely separated, you can make a completely different kind of interface. You can do a coacervation where you use charge to coat the surface with a very, very thin layer of an elastic shell that has no surfactant. So you can make emulsions which have no surfactant. You can make perfume without using alcohol because you can emulsify the fragrance molecules. There it's doing, and this can be scaled. This is actually a production unit, and this is actually something that Capsum does, and now you can buy it, and if you look carefully, a revolution in hydration powered by micro droplets. So this material sold by Chanel actually is made with microfluidic techniques. They made something like 1 million units in less than 9 months. So this, to me, is proof positive that not only can you do interesting physics and do interesting science, but you can actually make this in a very practical way once you understand something about the scale-up. You can make other things. We talked about food earlier. Here's some fake caviar. You can make a lot of different kinds of foods. So there's a lot of opportunities for making things. You don't have to be afraid anymore about scaling things up. And with that, let me finish, and thank you for your attention, and take any questions.
Is this microphone on? You know, I thought your skin was looking dewy and younger.
You know my tricks now.
I do. I would like a discount.
Me
Question
too.
over there.
Yeah, I'm building a micropipette aspiration device, and most of this equipment can be purchased on eBay by one person. And micro-injection technologies, Validine, pressure transducers, computers that can control pumps, and most flow rate can be controlled by one individual running something like LabVIEW. I'm just curious as to how a lab can support research. Actually, I have published most of this a few years ago in 2007. I actually wrote a poster on micropipette aspiration of a membrane cytoskeleton interaction. My questions are basically about the physics of the plaquette that makes up the surface of the membrane or the vesicle, and if the hexatic or pneumatic order, which is one of the phonon-mediated orders of the membrane, can actually support the pressure applied from micro-injection at 180 Mach number, which seems completely Well, I mean, it doesn't seem like we can get caviar to move at that speed and still maintain its structural rigidity because it's a very soft membrane. When I was working in Berkeley, I remember distinctly the capillaries, which each took about 30 minutes to make. These tubes are very difficult. I don't know if the 30-minute window you say of making a membrane micropipette aspiration device is necessarily correct. I'm kind of skeptical about that because you have to Teflon coat those surfaces as well. And you also have to monitor the pressure. The vibrations from the pump also coupled to the vesicles. That wasn't taken into account.
So it sounds like you're a real expert, and so maybe you should come and explain all the things that I got wrong. I'm sure I got a lot of things wrong, but I will say this, that my students and postdocs really can do this, and they can do it in half an hour. They can make it work. The fact that the Mach 160 was not to make vesicles. That's to make small drops. That's really something quite different. But I wanted to try and tell you a little bit about everything.
Yeah.
Thanks. We have another question over here.
Okay.
Thank you. Over here.
Yeah. My question is about the sort of range of sizes, especially the small size range, and monodispersity. And I was thinking about like colloidal gold nanoparticles that have certain specific selected sizes. I just wondered if you, and also coatings. I wondered if you have any comment on that.
Yeah, I mean, to make pure drops, the easiest size range is a few microns and above. To make the spray dry drops, we're down at 300 nanometers, thereabouts. But that's a little different. For making colloidal gold, I think if you're at 1 micron, using Faraday gold, you're at one particle per drop. You can just about do it, but it's tough. I mean, and the reason is just a practical reason. It's clogging. You have to really filter really well to prevent clogging. You can do that. You just have to make sure there's no dirt.
We can take one last question.
I kind of follow up on that. You told us not to be afraid. We're certainly not be afraid of electric or magnetic field. So I think it's possible if you kind of making those droplets in some external field, maybe you can control the, let's say, the size and the spatial distribution.
Absolutely. You can put an electric field across this. You can do take advantage of the charging of the drops and make much, much smaller drops. The problem with that is it's a little bit more difficult to control the size as precisely. It's also much more difficult to work with them. We've done a little bit of that, but not that much.
Okay, please join me in thanking Dave again for a beautiful lecture.